everyone. This is Stephanie Tosti, and I am the spokesperson for my group, which is the physical components of soil health group. And the different parameters that we measured were aggregate fractionation, bulk density, water holding capacity, and infiltration rate. Some background information which you will find in your lab manual regarding the field site or the experiment location is that the experiment was split into two sections, non-cover crop versus cover crop, and mono versus intercropping. Intercropping had squash, bean, and corn, which are better known as the Three Sisters. Once our group had all this background information, we then had to decide what our focus was going to be for our research. So our focus was taking samples of soil samples for the crop of squash. So we specifically just looked at squash and we compared non-cover crop to cover crop. And within a lab manual there is a video link that we recommend watching because it actually demonstrates how to do the steps that I'm going to explain. Step 2 would be that you need to split up the field area by location, so meaning decide where you're going to take samples at. You also need, after you do that, to get your tools that are needed. The tools that we needed were bags, which could be Ziploc or paper bags. We recommend Ziploc bags. The reason why I recommend Ziploc bags, they're a little bit sturdier. You could close them better compared to a paper bag, which you can't close. You just have to close with your fist. And Ziploc baggies could be potentially better to write on. So the next tool that was needed was a Sharpie or marker that you could write on, so you could write on the bag with. And you also need a trowel. It's like a small shovel that you'll need to use in order to take your soil samples. After you get all your supplies or tools rounded together, you will then need to label your bags by the location that you decided or how you split up the field. 5, you will need to take your samples. For our experiment, we took samples for both cover and non-cover crop for the squash crop only. We split up the fields of cover crop versus non-cover crop into location of direction. So we split it into south side, east side, and west side of the field. We took one sample per each bed of the where a squash plant was located. We dug 4 inches deep, which is approximately 10 centimeters, which is also listed in your lab manual. Our 6th step would be that after we collected all these samples, the TA would oven dry our samples. The following week, when next time we were in lab, we weighed, after the samples were oven dried, we weighed out our samples. We weighed out 40 grams of our samples. After we weighed out the 40 grams of our samples, still in the same lab, we filled up a container or tray with water. It was about 8 centimeters deep. 1 centimeter of water needs to be above the sieve. So you're going to be sieving the 40 grams of soil into this container that is now filled with water. You're going to sieve for approximately 2 minutes, which is about 50 times up and down motion. We recommend that you use a metronome to help you achieve the 2-minute or 50 time mark without having to count for yourself. After you have sieved, so that's the first step, you're going to rinse whatever is on the actual sieve plate or sieve mechanism with a wash squeeze bottle. This wash squeeze bottle needs to have DI water in it. This is called the backwashing process, which is again listed in your lab manual. And the backwashing will be put into a pre-weighed Ziploc bag. This step is repeated for large microaggregates to microaggregates. So you go from large microaggregates, large macroaggregates, small macroaggregates, and microaggregates in that order. Step 8 is that whatever remaining water you have at your last pan from step 1 from large microaggregates to the last step of microaggregates, the water that is in each pan gets dumped into the next pan. So in the third pan, you're left with a large amount of water. This remaining water is what's going to be oven dried by your TA. You are now at step 9, where you will weigh the oven dried samples. Step 10, or the last step, is to calculate your dry weight percentage. This is all the steps that you will need to do in order to achieve the parameter of aggregate fractionation. Some other suggestions that our Pacific group had for next year's class is to make sure that all your materials or all the tools that you have collected from beginning to end throughout your whole experiment don't have any holes or any issues with them whatsoever, so that they're functioning to their best of their ability, so you won't run into any issues later.
The water holding capacity was determined using the Saxton and Rawls equation derived from the baseline data, sand, clay, and organic matter percentages, and the bulk density samples, 4 from each system, obtained on the 3rd week. Using the baseline data, the saturated and field capacity were calculated to compute normal bulk density. Because bulk densities can fluctuate by location, we averaged the fallow and the cover crop bulk densities to determine the density adjustment factor to account for these variations. We were given an adjustment factor of 1.1 for fallow and 1.0 for cover crop by the TA. With the new adjusted bulk density, we recalculated the water potential at saturation and field capacity. In the final step, we subtracted water content at permanent wilting point, derived from the baseline data, from water content at field capacity. Our results showed that agrosystems that practice cover cropping have a higher water holding capacity than agrosystems that practice fallow.
Infiltration rate refers to the speed at which water at the soil surface enters into the soil. This test is beneficial in informing the farmer or researcher on how good their soil structure is. The better the soil structure, the more stable pore networks are, which allow the soil to capture water better, reduce runoff, flooding, and soil erosion. Our hypothesis for the infiltration rate test was that the squash with a cover crop would have a better infiltration rate than squash with no cover crop. After analyzing the test results, we concluded that our hypothesis was right. The test showed that on average, the squash with no cover crop infiltrated 500 milliliters at a rate of 47.53 seconds per 1 milliliter, and the squash with a cover crop infiltrated 500 milliliters of water at a rate of 1 second per 1 milliliter of water.
To determine bulk density, which is the amount of soil material in a given volume, we took a total of 8 samples using a 10 centimeter diameter by 10 centimeter length core, mallet, sampling bag, and a small 4x4. We drove the soil core downward into the soil profile and then dug around it and beneath and lifted the core into the sample bag. Then we weighed the fresh weight and then oven dried it. We weighed the oven dried and then we divided that weight by the total volume. Our results. We averaged our results and derived the standard deviation and calculated the t-test, which showed that they are not very different. That the bulk density of fallow and cover were not very different. There you go.
I think a really fun thing about this project is that we were able to kind of decide what actual experiments we wanted to do. We had a very specific group of what to experiment on, but how we were kind of going about that was more our own personal choice, and so I thought that was very fun about the project. So being part of the soil physical group, there's, depending on how you kind of go about it, there's not necessarily as much to kind of work on from a week to week, lab to lab basis, but we were able to get good information and really start organizing early, which I thought was a very, very crucial part to our group and to my personal success with this project, is really deciding, you know, how this is going to play out in my final project, and how, as we continue to go through the quarter, we can use the kind of the day-to-day stuff to write down notes and, you know, make a really nice collaborative final project. So for some advice for future cohorts, it's definitely important to really organize yourself and keep the data in line. Really kind of make mental maps of where you are working out in the field, and how each kind of individual part of your experiment is working together in kind of a spatial realm in the field. At least for me personally, that's very helpful to keep it all in track and kind of picture where it's all happening in the field and how it's going to all play together.